The Fore people of Papua New Guinea had a word for it. Kuru, to shake. It began with a tremor in the fingers, subtle enough to dismiss. Then the feet. Then the torso. Within months, the body could no longer stand. Speech slurred into silence. But the mind stayed. Patients remained fully conscious, aware witnesses to their own unraveling. Some erupted into involuntary laughter. When physicians arrived in the eastern Highlands in the 1950s, they called it the laughing death. No cure, no identified cause, no precedent anywhere in medicine. A pattern emerged that deepened the confusion. Women and children were dying at far higher rates than men. Some villages lost nearly every adult woman. If this were a pathogen in the water or the soil, why would it select so precisely along lines of age and gender? The disease produced no fever, no immune response, no inflammation. Brain tissue examined under a microscope looked as though it had been eaten away, riddled with tiny holes, like a sponge. But no bacterium, no virus, nothing. In 1959, a veterinary scientist named William Hadlow noticed something unsettling. The brain lesions of Kuru patients looked almost identical to those of sheep affected by Scrapie, a neurodegenerative disease studied in British agriculture for over a century. This shifted everything. Could Kuru be transmitted? Carleton Gajdusek took brain tissue from patients who had died and injected it into chimpanzees. After months of silence, the animals began to tremble. The disease had crossed species. Gajdusek received the Nobel Prize for this work in 1976. But what was in the tissue? Anthropologist Shirley Lindenbaum spent years among the Fore. She traced the disease to a specific funerary practice. When a community member died, relatives consumed the body in a ritual the Fore understood as an act of love. The dead would be absorbed into the living, not left to decay in the ground or be consumed by insects. The brain, the most infectious tissue, was handled primarily by women and children. That single cultural detail explained the entire demographic pattern. The practice ended in the late 1950s under Australian administrative pressure. And yet, people kept dying. New cases appeared in the 1970s, the 1980s, the 1990s. In 2006, researchers documented patients whose incubation periods exceeded 50 years, individuals exposed as children, a half-century earlier. The agent responsible was unlike anything biology had ever classified. In 1982, Stanley Prusiner identified it as a prion, a single misfolded protein. No DNA, no RNA. It replicated by touching neighboring proteins and forcing them into the same wrong shape. A chain reaction of corruption at the molecular level. It could not be destroyed by standard sterilization. It triggered no immune response. It was neither alive nor dead in any framework science had ever used. The discovery reached far beyond Papua New Guinea. Prion diseases were later identified in cattle and in humans who consumed infected meat. The same invisible mechanism that had hidden inside a mourning ritual had silently crossed into global food systems. The Kuru epidemic is now considered ended. Approximately 2,700 documented deaths over 5 decades. The last known patients died between 2009 and 2012. The Fore believed that consuming their dead was the most profound act of love available to them. The person you lost became part of you, literally, cellularly. Hidden inside that devotion was something no one could have predicted. Not a bacterium, not a virus, a single misfolded protein. A wrong shape that taught other shapes to be wrong. Every culture on Earth has rituals for the dead. We bury, we burn, we embalm, we preserve. Each practice carries a biological signature we rarely pause to examine. The Fore story is not a warning about one people. It is a demonstration that love and danger are not always opposites. Sometimes they share the same gesture, held for a lifetime, and then held longer still.